Have you ever really thought about how a cut on your finger heals, or how a single fertilized egg grows into, well, you? The secret to all of it is the cell cycle. It's basically life's most fundamental program for making copies of itself. Think of it as a cell's ultimate mission: duplicate everything perfectly and then split into two. So let's follow along on this incredible journey.
All right, so this diagram is the cell cycle in a nutshell. You can see it's this incredibly organized loop with two main parts: a really long preparation phase and then a much shorter, super dramatic division phase. All the prep work happens in stages. You've got G1, where the cell just gets bigger, then comes the really critical S phase, where all the DNA gets copied, and finally G2, which is like a last-minute check. All of that leads up to the main event, the M phase, where one cell finally becomes two.
So here's our game plan. We're going to follow the cell's mission right from the beginning. We'll start with its whole purpose, then see how it copies its genetic blueprint. After that, we'll dive into the amazing control system that makes all the big decisions. Then it's time for the really cool part, the dramatic split of the chromosomes, and finally, that magic moment where one cell becomes two.
So, you might be asking, why does a cell go through all this trouble in the first place? Well, for a little single-celled guy like yeast, this cycle is how it creates a whole new organism. But for complex creatures like us, it's the engine of life itself. It's what powered our growth from a single egg, and it's constantly working to replace old cells. But no matter the organism, the mission is always the same: pass on that genetic blueprint to the next generation and do it flawlessly. To pull this off, every single cell has to absolutely nail three fundamental tasks, and it has to be in this exact order. First, it's got to grow and, most importantly, replicate its chromosomes. Second, it has to accurately separate all those copies. And third, it needs to physically split its guts, the cytoplasm, in a process called cytokinesis, to create two brand new daughter cells.
Now, how does a cell budget its time? Well, it spends most of its life in what's called interphase. That's G1, S, and G2 all lumped together. This is the long, kind of quiet period of just growing and copying DNA. It's all just prep work for the M phase, which is way shorter but way more dramatic. That's when the action really happens and the division finally goes down.
Okay, let's talk about the most critical part of that whole prep phase, the S phase. This is where the cell takes on the incredibly high-stakes mission of duplicating its entire genome. The S actually stands for synthesis because it's synthesizing brand new DNA.
And yeah, this is not a quick job. In a typical mammal cell, the S phase alone takes about 10 to 12 hours. I mean, to put that in perspective, that's like taking an instruction manual with 3 billion letters, reading it, and typing out a perfect copy without any typos. The cell spends all this time making sure every single base pair is copied with just unbelievable accuracy.
Now, this next part is a really crucial distinction, so listen closely. Before S phase, our cells have what are called homologous chromosomes. Think of them like a pair of shoes. They're a pair, but they're not identical copies. You get one from your mom, one from your dad. But after S phase, each of those individual chromosomes has been duplicated. This creates a pair of sister chromatids, and these are perfectly identical copies, literally stuck together.
So, the main thing to remember here is that a sister chromatid is an exact replica of the original. These identical twins are fused together at this spot called the centromere, and that's what gives them that classic X shape you always see in diagrams. Seriously, understanding this difference is the key to everything that's about to happen.
Okay, with all of this unbelievably complex stuff going on, how in the world does the cell keep it all straight? How does it know when it's okay to move on to the next step? Well, it has this incredibly sophisticated internal surveillance system, kind of like a cellular mission control, that makes sure each step is completed perfectly before the next one even begins.
So yeah, who's in charge here? It is definitely not just chaos. There's a whole network of proteins that acts like the director of this entire performance, and we call this the cell cycle control system.
This whole control system operates at three major checkpoints, these go-no-go moments. The first one is latent G1, where it checks if the environment is even right for division before it commits to copying all its DNA. The second is at the G2/M transition, where it confirms that all the DNA has been copied and any damage is fixed. And finally, right in the middle of mitosis, it asks one last, crucial question. Are all the chromosomes properly attached to the spindle? Only then does it give the final green light to separate them.
So what molecules are actually running this whole show? It boils down to two main characters: cyclins and something called cyclin-dependent kinases, or CDKs for short.
The easiest way to think about it is this. The CDK is like an engine. It's always there, but it's switched off. The cyclin is the key. When the cell makes the right cyclin, its concentration goes up, it finds and binds to the CDK, turns the engine on, and boom, the cell is driven into the next phase.
And here you can see that core mechanism in action. You've got an inactive CDK enzyme, just hanging out. Then its specific cycling partner shows up and binds to it. This creates an active complex, which then acts like a switch. It goes around and phosphorylates other proteins, which is just a fancy way of saying it turns them on, to trigger all the key events, like starting DNA replication or breaking down the nucleus wall.
The whole system works like a molecular clock, and it's driven by these waves of different cyclins. You can see it on the graph. First, the G1/S cyclin levels peak, and that pushes the cell into S phase. Then, the S cyclins rise to drive the actual DNA duplication. And finally, M cyclin levels build up to trigger mitosis. And once each job is done, that specific cyclin is destroyed, which basically resets the clock for the next phase. It's so elegant.
But this system isn't just an accelerator. It also has a really powerful brake. If mission control detects a problem, like some DNA damage, it slams on the brakes by blocking those CDK complexes. This is so important because it ensures the cell always prioritizes accuracy over speed, preventing catastrophic errors before it's too late.
All right, so our cell has grown, it's copied its DNA, and it's passed all the safety checks. Now we enter the M phase, and honestly, this is the most visually spectacular part of the whole journey, mitosis. This is where the cell is going to meticulously divide its duplicated chromosomes into two absolutely identical sets.
And the star of this show, the main piece of machinery, is the mitotic spindle. It's this incredible structure made of protein filaments called microtubules. You can see there are three types that have very distinct jobs. The kinetochore microtubules are the ones that actually grab the chromosomes. The intrapolar ones push the cell's poles apart, and the astral ones just make sure the whole thing is positioned correctly.
The show officially kicks off with prophase. Inside the nucleus, those super long chromosomes start to condense dramatically, coiling up so tightly that they finally become visible under a microscope. At the same time, outside the nucleus, the two centrosomes start moving apart, beginning to build that mitotic spindle between them.
Next up is prometaphase, and it starts really abruptly when the nuclear envelope just breaks down. This is such a crucial step because it finally frees the chromosomes. Now, the spindle microtubules can reach in and attach to a special spot on each chromosome called the kinetochore. They're capturing them and getting them ready to move. All that commotion leads us to metaphase. After a bit of a tug of war, all of the captured chromosomes are lined up perfectly along the equator of the cell, on this imaginary line we call the metaphase plate. The cell is now in this state of super high tension. Everything is poised and ready. This is the moment of that final checkpoint, right before the irreversible step of separation.
Okay, so everything is perfectly aligned. This is the point of no return. But how does the cell coordinate this? How does it make sure that every single pair of sister chromatids separates at the exact same instant?
The signal comes from a machine called the anaphase promoting complex, or APC/C. Once it gets the all-clear signal, the APCC goes and destroys a protein called securin. Think of securin as a safety lock on a pair of molecular scissors called separase. So with the lock gone, separase is unleashed, and it immediately goes and cuts the cohescent proteins that's the glue that's been holding the sister chromatids together this whole time.
And the result is anaphase. As soon as that glue is gone, the sister chromatids separate instantly. They are now considered their own individual chromosomes, and they get pulled swiftly to opposite poles of the cell.
Anaphase is actually a two-part process that works perfectly in sync. In what we call anaphase A, the microtubules reel the chromosomes in, pulling them towards the poles. At the exact same time, in anaphase B, other microtubules are pushing the poles of the cell themselves further apart. This two-pronged attack ensures a really clean and complete separation.
Okay, the chromosomes have been successfully segregated. The cell is now long and stretched out, with a complete set of genetic instructions at each end. We're in the final act now, where the cell finally completes its mission of creating two new, fully functional daughter cells.
This kicks off with telophase, where basically all the events of prophase are run in reverse. The chromosomes arrive at the poles and start to decondense, becoming long and stringy again. A brand new nuclear envelope reassembles around each set,
creating two new nuclei. And with that, mitosis, the division of the nucleus, is officially complete. But the cell still isn't quite done. Overlapping with telophase is cytokinesis, which is the division of the cytoplasm. A contractile ring of proteins forms right around the cell's middle, and it tightens like a drawstring on a hoodie, pinching the cell in two and finally completing the creation of two separate, identical daughter cells. So, just to put all of this in context, what we've just watched is mitosis. Its whole goal is to create two genetically identical diploid cells, which is perfect for growth and repair. Now, this is very different from meiosis, which is the process that makes sperm and eggs. Meiosis is a two-step division that produces four genetically unique haploid cells. And the biggest difference? In the first part of meiosis, it's the homologous pairs that separate, not the identical sister chromatids.
The cell cycle is just this beautifully precise program, absolutely essential for life. You've got the checkpoints, the timers, the spindle. It all works together in perfect harmony. But this brings up a really crucial question. What happens when this code breaks? When that control system fails, cells can start to divide uncontrollably. And that, unfortunately, is the basis of cancer. Understanding this fundamental cycle is really the first step in learning how to fight it.